Now, for section 3, we're going to be looking at what it takes for a virus to multiply. For all intents and purposes, when it comes to viruses, one goes in, thousands, hundreds, thousands, millions come out. Well, how do you go from the one to the hundreds, the thousands? Well, turns out there are 5 generalized steps that we're going to be looking at over this lecture and the next lecture. 5 steps: adsorption, penetration, uncoding, synthesis, assembly leading to release. Okay? You'll need to know these 5 steps. What we're going to do, in case you haven't already guessed, is we're going to go over each of these steps individually. So when you look at this figure here, this figure summarizes those 5 steps. Now it's a little hard to see on the slide, and it's a little hard to see in your handouts, so I suggest you go to the textbook online and look at table 5.4. Okay? You'll notice it has the 5 steps listed out. Adsorption means attachment. The spikes that we talked about in the previous lecture, those somehow have to interact with the receptor. Interaction of the spike to the receptor is going to trigger 2, penetration. Somehow the virus has to get inside the cell. Whether it's phagocytosis, the cell engulfs, or if it's an enveloped virus, the envelope fuses to the plasma membrane. This will release the nucleocapsid into the cytoplasm. Once there, the uncoding, basically the capsid opens up, releasing the DNA or the RNA, leading to step 3, synthesis. The making of copies of the genome, the making of the capsomeres. Assembly, well, exactly what that sounds like. Getting the capsomeres to self-assemble around copies of the genome. And then release. Somehow the newly formed nucleocapsids have to get out. And we'll talk about these different steps. So, the absorption. Turns out all viruses have what they call a host range. Okay? Host range means that we, as humans, mammals, we're constantly exposed to viruses that are for fungi, for plants. But because we're not the correct host type, the spikes on those viruses cannot interact with the receptors on the surface of our cells. Now keep in mind, receptors, think of these as glycoprotein antennas. They stick out from the cell, and they respond to one and only one signal that may be floating by in the bloodstream or floating by in the interstitial fluid. Over the millions of years of evolution, viruses have evolved to hijack our receptors. So when it comes right down to it, you do not have an influenza receptor. You do not have an HIV receptor. What you have are receptors for other molecules that HIV, that influenza, all the other human viruses have hijacked. So the example from the textbook, hepatitis B. Hepatitis B, human only. Okay? It's not going to be able to infect your dog. It's not going to be able to infect your goldfish. It's not going to be able to infect an insect. Its host range is us. Hepatitis B, its spikes only react to glycoproteins on the surface of certain liver cells. So the viruses are restricted not only to host, but certain cells in the host. They refer to that as tropism. Now, once the spike interacts with the receptor, you're going to end up with what is referred to as penetration occurring. Okay? Penetration. Somehow the virus has to get in. Okay? It may be the entire virus, or for some viruses, it's just the genome. They stay outside the cell, the capsid stays outside, and the genome, the DNA or the RNA, somehow is pushed into the cell. Multiple methods all end with the genome being in the cytoplasm. So if it's one of these viruses where the entire nucleocapsid goes in, well, then you have to have the encoding step where the capsid breaks down into individual capsomeres, releasing the DNA, releasing the RNA. You look at this figure from the textbook that's behind my head. You'll see that spike interacts with the receptor. It triggers changes in the cell membrane. It triggers the cytoskeleton to rearrange, cause the membrane to start moving about. By moving about, it'll cause interaction from cell membrane, envelope. They interact, they fuse, which is what you see in the part of the figure above my head. Sometimes instead of interacting and then fusing, they'll interact and then engulf, as you see over there to the far side. Either way, once the nucleocapsid is in the cytoplasm, it will have to break down. It will have to release the genome. Are there exceptions to this? Yes. There's exceptions to all the rules when it comes to viruses. But like I said, this is the generalized method for absorption and penetration.